So in our group, we work with bioengineered microenvironments to promote cell assembly in 3D under controlled conditions. So we have established different types of microtissue systems that recapitulate tissue-specific morphogenesis and differentiation, and we are trying to understand the impact of the microenvironment on such processes. So we have been developing molecularly designed hydrogels with tunable biochemical and biomechanical properties, which are cell instructive, and so we can use them to guide cell response and organization in a very controlled way. So we believe that these systems are very useful tools to understand how cells sense the microenvironment, to organize into specific structures, and also to investigate the role of the extracellular matrix and also of other cells in morphogenesis. So ultimately, we would like to translate this knowledge into the development of more effective tissue regeneration strategies and also into the design of advanced models, so 3D models for in vitro studies, which could be easily applied in different fields of research, from regeneration to cancer. So, well, when I'm not working, I like to be with my friends and family, this big priority, and I also like to read, to travel, and also I do some Pilates and yoga to calm down my body and soul.